So let's get on to our last topic here, limitations on ATP yield. We'll talk about lipid metabolism, and we're going to talk about how you regulate ATP production. So limits on ATP yield. Let's start with what we know already. We know that we go from a glucose and that we end up with 2 pyruvates during glycolysis. Out of that, we get 2 ATP net. Remember, we put 2 in, but we got 4 out of glycolysis. And we also got out a couple NADHs. Okay, these are our cytosolic NADHs that we talked about in the last lesson. So we go through glycolysis, then the pyruvate. If you have oxygen available, you transport the pyruvate into the matrix of the mitochondria. If you don't have oxygen available, the pyruvates will run through fermentation. So keep that in mind. This is the aerobic pathway when oxygen is available. So what do we get? Well, from our pyruvate, 2 pyruvates convert to 2-acetyl-CoA. So we go from 3 carbons to 2 carbons because we chopped off a carbon dioxide per pyruvate. And we captured another couple of electrons. Those acetyl-CoA enter the Krebs cycle. They crunch together with oxaloacetate to make citric acid. And then we run through that twice, and we end up getting our 3 new goose feathers twice, which is 6 NADHs, a couple GTP that become ATP, and then we get our 2 FADH2s that are coming out of that. Keep in mind that every time, well, maybe not every time, keep in mind that when we created the first 2 NADH, as we created here and created here on the cycle, that each time we chopped off the CO2, we were able to capture electrons. And it wasn't until the very last one, from malate into oxaloacetate, that's the only time that we've made an NADH in the Krebs cycle and didn't have to do any cutting off of a carbon. Okay, so we cut off a carbon dioxide, make NADH. Cut off carbon dioxides, make NADH. Keep that in mind. So anytime you cut off a carbon dioxide, an NADH is made. That's really a good thing to try to remember. Okay, what do we end up doing with all of these electron carriers? Well, we carry them to the electron transport chain. Keep in mind, this is on the inside of the cristae, or the inside of the on the matrix side of the inner membrane. So we're going to take these cytosolic ones from the outside in the cytoplasm. So we're in the cytoplasm versus matrix. And cytoplasm, we're going to give them to either the glycerol phosphate shuttle, the GP shuttle, or give them to the aspartate shuttle. They're going to pass them to either an FAD to make FADH on the inside, FADH2, pardon me, on the inside, or an NAD on the inside to make another NADH. Then I get 4 to 6 ATP out of that. From these 2 NADHs, from the oxidation of pyruvate, I'm going to get 6 ATP value. They're already inside, so I get full 3 ATP per NADH for this one. Full 3 because they're on the inside for these NADH, full 2 for the FADH2. And of course, I've got my oxygen coming in, stealing electrons from the chain and forming water in order to backwards drive all of that. So in total then, I had 2 ATP produced through the substrate level phosphorylation here, another 2 through substrate level phosphorylation here. So a total of 4 net by substrate level phosphorylation. Memorize this number, 4 ATP net by substrate level phosphorylation. The rest of them, 32 to 34 ATP, are made by oxidative phosphorylation. Make sure you really understand the difference between oxidative and substrate level. You're going to be asked to give me those definitions and distinguish between them. So make sure that you are confident you can do that. So then as a total from 1 glucose, I should get 36 to 38 ATP. The problem is that I don't actually get 36 to 38 ATP. The actual values of ATP produced are actually lower than this per glucose. And there are 3 reasons I'd like you to memorize. The first is that some protons leak through the inner membrane without passing through ATP synthase. So if we go back to our other diagram, recall that we had the protons being pumped out with the 3 different pumps from the matrix into the intermembrane space where they weren't allowed to escape. And we were hoping that every proton would just flow back through ATP synthase and make me some ATP. But if some of these protons leak back through the inner membrane without going through ATP synthase, then I get no value of ATP off of this proton. The only way to get it to have some value is if it goes through the ATP synthase. So I don't make full ATP. I don't make the 36 to 38. For one reason is that these guys might leak through here rather than going through the channel that I want them to go through. Okay? The second reason is that some of the energy from the proton motive force, that term was introduced in the last note, proton motive force is used to transport the pyruvate into the matrix. If you want to get the pyruvate from the cytoplasm through the outer membrane, through the inner membrane, and then into the matrix, you're going to have to pump that sucker. So the two pyruvates are going to have to be pumped into the matrix, and that's going to require a little bit of energy. It doesn't require ATP to do that. It requires proton motive force. So that's when the protons are flowing, pyruvate takes advantage of that and comes in. The same thing, proton motive force is used to transport the ATP out of the matrix as well. So if I need to have all these ATPs produced inside, I'm going to need to get them back out to the cytoplasm where they can actually do some good for the rest of the cell. And if I want to get them out there, I'm going to have to pump them across. And again, I do not pump them by using an ATP. I pump them by using protons to open channels that allow the ATP out. But every proton that I use to either move pyruvate in or ATP out is a proton that doesn't go through ATP synthase. So that's why it's going to reduce our total from 36 to 38 to something more like 30 to 32. So even though a normal NADH is supposed to give us the value of about 3 ATP, on average, it actually gives us more like 2.5 ATP. And every FADH2 is supposed to give us 2 ATP, but on average, they give only about 1.5 ATP. So keep that in mind. Please memorize this number as well. 30 to 32 ATP per glucose is more practical, although this 36 to 38 is ideal. The question you're going to be asked is something along the lines of, why do the actual values of ATP produced, why are they lower than the 36 to 38? And you're going to give me these 3 reasons right here. Our next topic, lipid and protein metabolism. What is that? Well, metabolism is when you're breaking stuff down for the purpose of creating energy. Now, we've just gone through carbohydrate metabolism when we talked about all 4 stages, the glycolysis being the first. We get our pyruvate. Once we have pyruvate, we're able to go through oxidative respiration, and then we're able to run through the Krebs cycle, and then get the ATC. So this here is our regular carbohydrate metabolism, when you have glucose and you run right through. But I think you already know that you can burn fats and proteins for energy if you want to. And the way that you do that, you can even technically use nucleic acids, although if you're using nucleic acids for energy, you're in big trouble. So instead of using just carbs and the monosaccharides, which could be converted to the monosaccharide glucose, you can take the protein. Remember that protein is composed, its monomers are called amino acids. So big protein is a whole huge long chain of folded amino acids, both alpha helix coils and beta pleated sheets. Well, different amino acids, these are just 4 of them here. Remember, there's 20 in the human body. Alanine, leucine, proline, and glutamate. Depending on which amino acid they are, remember they have a different R chain on them. You have the regular NCC backbone of an amino acid, but then dangling off of that middle carbon, you're going to have the different R chains. And that's what separates alanine, leucine, proline, and glutamate. So depending on which one they are, they can be converted to different molecules in the chain. Alanine can be converted to pyruvate and enter the chain here. Leucine can be converted to acetyl-CoA. I have forgotten the L again. This is insanity, but whatever. It's all good. You guys know there's supposed to be an L there. Proline and glutamate can enter at different points in the Krebs cycle. So I can convert these amino acids into something that can enter the chain at these particular points. Notice I'm able to do that with acetyl-CoA here. Now, with fats, remember that fats do not form polymers. Instead, they're formed by having a glycerol. There was a carbon, 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 and you had the 3 chains coming off of them. It's formed by a glycerol with the 3 fatty acid chains. And I can separate, when I digest the fat, I can separate those guys out. The glycerol already has 3 carbons. So it can be very easily converted into a glucose molecule, just like G3P had the 3 carbons, and it was converted into a glucose molecule back when we were talking about photosynthesis. So the glycerol goes through something called gluconeogenesis. The word neo just means new. So this is the creation of new glucose. Right? So from the glycerol, very easy to do that. I can also convert glycerol to DHAP. If you remember, in the middle of our glycolysis, right when we were splitting the glucose for the first time into 2 3-carbon molecules, one of them was a G3P and the other one was a DHAP. The DHAP had to be converted to G3P to run through the program. So here we are. If we can make the DHAP, we already have the machinery to do this conversion. It's very easy to slide it in there. If it's the fatty acids that I'm looking at, then the fatty acids can be chopped up. These are big, long carbon chains, hydrocarbon chains. I can chop them up into 2 carbon molecules, and those are acetyl groups. So the acetyl groups, I just attach a CoA, and boom, I can come right into the process right here. So I can, as long as I'm in the mitochondria already, I'm in the matrix, I can just pump these guys in, a whole bunch of acetyl CoAs, and I'm ready to run through the Krebs cycle. If you want to see a better version of this, look at page 131 of your new text, and you'll be able to have a much prettier version of what this is here. So just a comment then, again, on protein catabolism. How does it work? Well, remember that, again, you've got this backbone that's identical for all of the amino acids and then the dangly R chain. Once you remove the amino group, which is called deamination. Now I have spelled this incorrectly here. This should be the M and the N need to be switched. So amination. So the removal of an amino. So again, please switch this N and this M. Must have been going too quickly when I was creating this. So deamination, not deanimation, of the amino acids. You get the NH3 gets removed. NH2 is removed and the other hydrogen joins on. Then I can take this 2-carbon molecule, and that acts as my acetyl group. And I CoA on the end here, and then I've got my acetyl CoA. Okay? With lipid catabolism, again, the fats get broken into glycerol and fatty acids. The glycerol gets converted to glucose because it's already a 3-carbon molecule through gluconeogenesis, or it can be converted to DHAP and then to G3P, like I said above. And then the fatty acids get broken down into 2-carbon molecules. Those are acetyl groups. Recall that this is what our acetyl CoA looks like. There's the acetyl group with the CoA on there. So as long as I can get these 2 carbons from somewhere, all I have to do is add a coenzyme A, and I'm good to go. Very easy to make an acetyl CoA from a fatty acid. Here's an example here. So lauric acid is quite a long fatty acid chain. It's got about 12 carbons. That's why it's called lauric, 12 carbons. Which means if I was to chop it into 2 carbons each, I would get 6 acetyl-CoA out of that. Now, it does require some energy, so it's going to use 1 ATP for every cut it makes of the fatty acid, but it's going to produce 1 NADH and 1 FADH2 for every time it does that. So it far outweighs the 1 ATP being used, hardly compares to the 1, 2, 3, 4, 5 ATP that I'm going to get. So I'm going to get a net of 4 every time I chop this. So 4 times the 6 gives me that 24 ATP advantage. Massive, massive, massive advantage over just using 1. So that's why you're going to see that fats actually end up giving us a higher energy yield if you were to burn a fat, 38 kilojoules per gram, than a carb. So pretty much just over twice as much energy in a fat as there would be in a carb, regulating ATP production. So your cell actually keeps a constant level of ATP. It can speed up or slow down production depending on how much ATP you're using. So if you are at rest, you don't need as much ATP as if you're in the middle of an exercise and you're burning through it like crazy. So you want to be able to control and regulate your ATP production. So how do you do that? Well, there's control in quite a few levels. The first bit of control we see is in glycolysis. Now in glycolysis, we didn't mention the names of the 10 enzymes that are involved, but if you wanted to go from glucose to fructose 6, remember we had glucose, then glucose 6-phosphate, and then we popped that carbon out and we got that fructose 6-phosphate. Well, in between the fructose 6-phosphate and then adding in that extra during secondary activation, adding in the extra phosphate to make fructose 1,6-bisphosphate, there was this enzyme right here, phosphofructokinase. A kinase is an enzyme that does the job of ripping off phosphates from ATP. So kinase enzymes do that. So if it's a phosphofructokinase, it's ripping off a phosphate group from the ATP and giving it to the fructo, or the fructose 6-phosphate gets another one. Okay, so this guy, very important. It's an allosteric enzyme that I'm able to inhibit. Now, I've listed it up here for you. Got an allosteric binding site where ATP can attach and non-competitively inhibit the enzyme. Okay, so you can have ATP being produced. If I've got a ton of ATP, then I really don't need to run through glycolysis at all. I may as well stop it before I waste any more energy. So it can come back and it can stop it. I can also kickstart it as well. So let's say I begin to run out of ATP, it stops inhibiting, and if I've got a lot of ADP available, it means my ATP is low. So ADP is able to turn this one on, as well as citrate levels. Now, if I've been able to go all the way through here and I've gotten down to citrate, if I've got a lot of citrate being produced, then I really don't need this running anymore because I've got enough citrate. So I want citrate, high levels of it, to be able to turn this off. But when citrate is low, I want the low levels of citrate to be able to turn it on so that I can have regulation during glycolysis. So the speed of glycolysis can be controlled by ADP and ATP levels and by the citrate levels as well. Then we get control in the oxidation of pyruvate. The pyruvate dehydrogenase, this is the enzyme that was doing the job of making the NADH and stealing electrons from the pyruvate. So if I've already got a lot of NADH, because that's the product of this pyruvate dehydrogenase, then I might not need any more. So if I have a lot of NADH, I can go back and shut this guy down so I stop producing NADH in the matrix. And that's the basic idea here. So if I'm producing NADH, producing, whether it's in this stage here where I'm going through pyruvate into the acetyl-CoA, or later on, this is the pyruvate dehydrogenase that we're talking about here. So the pyruvate into the acetyl-CoA, the NADH that's produced here can go back and inhibit and prevent this guy from running through the Krebs cycle. Then finally, in the Krebs cycle itself, some of the enzymes, we're not going to go through all their names because there's more than one. I don't want to overload you. Phosphofructokinase and pyruvate dehydrogenase are enough to remember. But some of the enzymes in the Krebs cycle are also inhibited by lots of ATP, lots of NADH, or lots of acetyl-CoA. Because these are such important end products of each of their stages, they're very good molecules to be able to go back and shut off the actual process. This is what we called feedback inhibition when we were talking about this in the first unit. Now, if you want more information, you want the written part of the text, or to see a better version of this picture, go to page 113 of your new text. And that, my friends, is the end of this lesson.